morning, and thank you very much for the kind introduction and for the invitation to come here to beautiful Yeonsu and giving me the opportunity to present some of my work to you. Also for the opportunity to, for the first time in my life, experience 24 hours of daylight. And I have to tell you, last night when I was trying to go to sleep, I experienced it more than I had wished for. So I'm going to tell you today about cortical mechanisms of color vision. And why cortical? Well, color vision basically can be thought of as consisting of several stages. In the first stage, we have three types of cone photoreceptors that transduce light into electrical signals. And these signals are converted into opposing colors in the retinal ganglion cells, still in the eye. And in the visual cortex, then, the excitation pattern of the cone photoreceptors are interpreted as colors. Now, we know the first two stages here we know a lot about. And basically what I've written here that practically everything is known about the retina. Of course, people who work on the retina will strongly disagree with that. But they've done a remarkable job. And it's really stunning what we know about color at this early stage. And already from this formulation, excitation patterns are interpreted as colors. You can imagine that we don't know very much about how this process works, but I'm going to hope that I'll be able to tell you a little bit about it. Now, of course, one of the things we know about is our cone excitation. So if we have light that gets reflected from an object, for example, from this red patch in the color checker chart here, we can measure the spectral distribution. And since the absorption spectra of the three different types of photoreceptors, the blue, green, and red cones, are known very precisely, we can just multiply these curves and then compute how much each type of cone is excited. And this process represents the two laws of early color vision, trichromacy and univariance. There's exactly three types of photoreceptors, and the excitation pattern of these three receptor types, these three numbers, are sufficient to characterize the effect of this whole continuous distribution of wavelengths on our visual system. And one of the reasons why the three numbers suffices is univariance, because the cones simply count the photons and they don't care what wavelengths it is, at least not directly. And given this, we can calculate basically for everything we see the cone excitations, how much are the red, green, and blue cones excited, and then we can do that, for example, for these four patches here, and you will see that the numbers we get, there will be sort of more blue cone excitation here, a little bit more red here, and most importantly, we can say, well, these are different. These lead to different excitations. Now, why am I saying this? This is trivial, and you all know that. Well, it gets more problematic if we introduce a context. Now, the central four patches look very similar to the ones I showed you just before, sort of this blue, yellow, green, and reddish-looking light. But what's happened here is that through the context, basically, and because the visual system is not looking at these patches isolated, it's looking at the whole region around it, four patches that would simply look gray if I presented them to you in isolation appear very colorful here. So these patches here are, in fact, they're really sort of equivalent to a gray if I would present it on a neutral background. That's why I've put this gray stripe here, but of course the stripe starts to look sort of more greenish here and more reddish here. But all I can do is to reassure you that these are indeed four identical patches here, even though they look very different, look more like what you see here. So for this type of analysis, we need to take larger regions of space into account, and we need to take sort of borders between colors into account, and basically that's one of the things that's happening in our brains, in the visual cortex. And that's not the only thing. This is a schematic presentation that I've taken from an article by Grusser and Landis, they've written a while ago, where they summarize basically how color sort of would affect visual processing. And of course, there's some retinal stages and some cortical stages, and basically I have colored those circles and rectangles. Basically, what we know well, I've used red tones for, and where we don't know anything about, I've given it a blue tone. And you can see that sort of at the retina, we know, as I said before, pretty much everything. And here, at the first stage of visual processing, we know a bit. Sort of we know something about how color is, the color of surfaces, and how colored borders are extracted. That we've learned a lot about this in the last 10, 20 years. But then when it comes to higher level effects of color, for example, the emotion, we know very little, and there are hardly any studies on that. And there's only, when it comes to these higher levels, there's been a bit of work on color categories and how we use color names. But other than that, this is all still blue areas. Okay, so what I'm going to talk to you about today is this stage of processing that basically happens in early visual cortex, the first cortical visual areas, primary visual cortex, secondary visual cortex, V1, V2, and so on. But before I get to the visual cortex, I'll just briefly like to go over these two stages that we know so well. And just to get back to the cone absorption spectra, which you can see here, there's two remarkable things. One, of course, is that the absorption spectra are actually quite broad. Especially the middle and long wavelength sensitive cones, which are sometimes just referred to as red and green cones, absorb light over the whole, almost the whole visible spectrum. But what's really remarkable and noticeable at first sight is that the spectra of the L and the M cones are so similar. They overlap to a large degree. And the reason for this is also quite well known now. They evolved only very recently. Well, 35 million is very recently in evolutionary terms. And the genetic basis for this is now also quite well known. And we know that the genes that code for these pigments are also quite similar in the DNA sequences. Now, this has the consequence basically that whenever somewhere in the visual field there's a light that excites an L cone, sort of a neighboring M cone will also get excited close to the same level. So we have a correlation between these two signals. And basically what I'd like to do is I'd like to show you two effects of this similarity. One thing is a very nice demonstration that Hans Brettel, François Vienneau, and John Mullen came up with many years ago now. They've taken this picture of flowers and they simulated what this picture would look like to persons who lack one of the cone types, either the L, the M, or the S cones. And basically this happens very rarely, but about 5% of the male population are affected, are they red-green colorblind or anomalous. And you can see here that there is color left in these images. These people are not colorblind, they're red-green colorblind. So they miss one dimension. But the two images are quite similar. And that similarity comes from the fact that the cone absorption spectra of the L and M cones are so similar. Another consequence is shown here. And what I've done here was I went to the local supermarket and bought some fruits and vegetables and put them on my terrace, measured them with a spectroradiometer, and then basically computed from the spectral measurements the cone excitations in the red cones or the green cones. And you can see that basically all the data points here fall on the diagonal. That is, basically whenever an L cone is excited by the light coming from a certain object, sort of the M cone that's sitting next to it will be excited almost the same level. So we have a very high correlation here, and that's not a very good way then to transfer the signals from the retina through the bottleneck of the optic nerve to the visual cortex. So we want the information processing to be more efficient. And Gershom Buxbaum was the first one who really thought about this in computational terms, and he thought, well, an efficient way to get rid of this correlation would be to do something similar to a principal component analysis. That is, we just look for two new dimensions. We don't take the L cone and M cone, but for example, let's take the sum of the L and M cones, which is basically here this luminance or intensity axis, as our first new dimension, and then there's a second axis, one that's orthogonal to it, and where we get the difference between the L and the M cones. And that can be shown to be quite optimal from a mathematical point of view. And the question then is, of course, well, is this what our visual system actually does? And it has been shown later, mostly by John Krauskopf and colleagues, that first psychophysically and then also in physiology experiments, single unit recordings from the retina or the visual part of the thalamus in monkeys, that this is indeed the case. And here are some experiments that John Krauskopf and colleagues did on adaptation. And adaptation is a psychophysical method to figure out what channels are used, or to possibly figure out what channels are used by the visual system to convey information to visual cortex. So mathematically, we have already established these color opponent channels, and what Krauskopf then did, he used an adaptation experiment where basically I've shown you the stimuli here in this color circle with an axis that's approximately sort of from red to green, and one that's where the S cones are stimulated differentially. And what Krauskopf presented his observers with large fields of light, and he measured detection thresholds. That is, the lights were modulated slightly, for example, in a certain direction of color space. For example, this one here, and the observer had to say whenever they saw something, whenever they noticed a modulation. And when you do that without anything else, you might get thresholds that are sort of on this circle here. And the distance from the origin basically presents the modulation depth of those light. How intense was the light? Now, when you adapt the observer before measuring those thresholds by sort of presenting a large modulation for 30 seconds, so it's going from red to green for 30 seconds, then that's shown here. We have this adaptation here on this axis, and you can see that the thresholds for detecting stimuli that are modulated on the same axis, also in red and green, are now much larger. So the sensitivity of the visual system in that direction decreases. Because, I mean, you can think about it as that channel sort of being tired. That's sort of not, it's actually not a good way to think about it, but it's sort of, it predicts what's happening quite well. So, but interestingly, if we measure thresholds in this other direction in color space, the adaptation didn't have any effect. So from that we can conclude, and from the results, from the analogous results in the other direction here, we can conclude that there's at least two channels conveying this type of information in our visual system. And the question here, and you can already see that these two channels are independent, sort of adaptation along one axis doesn't affect the other. The question is what happens when you sort of adapt in directions in between, that is now to something that's sort of from orange to blue. And if we have many of these channels, not just two, then we would expect sort of these elongated threshold contours here as well. Whereas if we only have these two channels from before, they would both get adapted by this intermediate stimulation. So if you stimulate here, then basically projecting this on the red-green axis or on the S-cone axis, both channels would be adapted. And thresholds basically would increase in all directions. And that's exactly what they found in their experiments here. So you don't get selective adaptation with a sort of a very elongated contour, but you get sort of similar adaptation in all directions of color space. So this means that these mechanisms are really the only ones There's only two here for the chromaticity diagram and another one for luminance. So there's the three color opponent, or two color opponent and one luminance channel, as was already suggested by the correlation analysis. And John Krauskopf didn't stop here. He went on to do experiments with Pete Lenny and Andrew Darrington, where they recorded from individual neurons. They not from color opponent cells in the retina, but cells at the next stage, sort of the lateral geniculate nucleus, which conveys visual signals to visual cortex. But the properties of these cells in the LGN are very similar to the properties of the retinal ganglion cells. And what's shown here is sort of the preferred color of each cell as given as an angle in this color space that I've shown you before. So red is 0, this yellowish axis is 90 degrees, 180 degrees greenish, and 270 degrees sort of the S-cone isolating axis. And the y-axis specifies sort of how much luminance each cell preferred. But that's not really very important now. But what you see here is that each data point, which represents that individual cell's preferred color, sort of falls in one of these four clusters. So the cells prefer the 0, 180 degree direction and the 90 degree, 270 degree direction. There's no cells or hardly any cells that are sort of in between these gaps here. So these cells in the retina or in the LGN are really the neural substrate for these so-called cardinal mechanisms of color vision that were shown here in these psychophysical experiments. So to summarize this retinal stage, basically there's three channels to the brain. And these color opponent channels are functionally independent. They're based on different anatomical substrates. There's retinal anatomists and physiologists have identified that sort of different types of cells really convey the information in these three channels in the retina. And they can explain quite many different color phenomena, but certainly not all of them. But this is basically the chromatic input to visual cortex. And I've summarized it here again in this diagram, which basically shows you this stage with the cone photoreceptors here, an image, how it, and coded the magnitude of the signals in the L, M, and S cone. And again here, this is very similar. And then followed by a color opponent stage with these three channels that lead to these very distinctly different channels. And of course, this stage sort of has been postulated and forwarded by Helmholtz quite a bit, and the second stage basically was favored by his opponent Ewald Hering, and the two of them were really going at each other with quite some vigor. But in the end, as often in science, it seems that both of them were correct in some ways, and that the true answer is somewhere a mixture of the two. They were both wrong a little bit. Helmholtz thought that the cone absorption curves would be sort of evenly spread out with wavelengths, and Hering thought that basically these opponent colors would be sort of what we could sometimes call the unique hues. So what you see here, and what's this image that shows how this is seen by the S minus L plus M channel, that's sometimes called by visual psychophysicists or the blue-yellow channel. And you see that it's not blue-yellow at all. It's sort of this greenish yellow and a very purple. And according to Hering, this would be colors that more similar to those in the Swedish flag. So the difference is shown here. Sort of these cardinal directions are quite different from what's called the unique hues. I've sort of made unique hue judgments here, and you can see the red is not far off, but the green is way off, the yellow is way off, and so is the blue. So this is very different. And basically, if we talk about the stages of color vision, then it has often been mistaken and also written in textbook that basically this retinal color opponent stage would be the basis for our unique hues and those sometimes called hearing colors. That's not true. That's something that must be happening at a much later stage, at a very high level of visual processing, where we have a small number of categories that are sort of tightly linked to language and memory, which are also, of course, very high level phenomena. But now we've seen what's happening here, and I've told you a little bit about these categories, but now we want to see what's happening here in between in primary or in secondary visual cortex with those color signals. Now, if we talk about cortical processing of color information, I have to tell you about the most prominent theory over the last 30 years or so. It's also a very simple theory. It's the coloring book theory of visual perception. And the way it works is basically, like the name says, basically you have the world, and then it's first analyzed and analyzed by a luminance-based system that basically draws in the edges. And that's only based on luminance because that can be much quicker. It's a much quicker process. And then only sort of sometime later, sort of the color vision system comes and sort of fills in the surfaces with color until we sort of have this real nice colorful image or percept of our world. And then, of course, if due to some brain damage, what can happen is that the color system fails, and then we end up with a perfect black and white rendering of our visual environment. Now, that was sort of the dominant theory for a very long time, and its major advantage was that it was very nice and simple and intuitive, but it wasn't really supported very well by data. And over the last, I guess, 10 or 20 years, there's been lots of evidence has been amassed that really show that it's not like this. And I'm going to present some of the evidence now that comes from physiology, from recording from single neurons in the brain of an animal, like a macaque monkey. There's also lots of evidence coming from psychophysics, but that would be a whole other talk. Now, what we did in our physiology experiments, and those were done a long time ago when I was still in New York, we simply determined the tuning properties of individual neurons. And those neurons basically come from the secondary visual cortex, an area that's quite accessible from the surface, and that played a very central role in this theory of segregation, basically where you have color analysis separate from the analysis of visual form. And what we did was we looked for individual neurons, and then we basically measured everything we could for each individual neuron. So, for example, we measured the orientation selectivity by presenting sort of stripes of different orientations. And then typically, if the response, here are the different orientations, here's the response in impulses per second. And when the response is much higher for one orientation than for most of the others, then we would say that that cell is orientation tuned, such as is the case for this cell here. And the first ones who discovered cells like that in visual cortex were, of course, Hubel and Wiesel in the 60s. And we can also measure the color tuning by presenting stimuli of different colors. And now again, if the response to one color is bigger than for most of the other colors, then that cell would be called color selective, because it could, in principle, signal colors. And we also look at how large the response to the best colorful stimulus was compared to the response to a luminance stimulus, sort of a white stimulus with the same luminance contrast as the colored stimuli. So when we looked at then for how the orientation selectivity of a cell would be related to the color responsivity, we found that basically the two are not correlated. So if there was a segregation of color and luminance, of color and orientation, color and form, we would expect that basically all the cells that are very selective for orientation, which is basically all the cells that are to the right of this line, would not show any color selectivity, that they would all be here at the lower right quadrant. And the color cells should all be in the upper left quadrant. And this is clearly not the case. There's no correlation here. And what you can see also is almost all cells in visual cortex, as has been known before, are orientation selective. But there's also about 50%, about half of the cells that we recorded from were color selective. And that's important because in many of the early studies, like Jubel and Wiesel, the proportion of cells in cortex that they classified as being sensitive to color was much lower. It was as low as 10%. But they only, basically they tested their cells with luminance, and if the cell responded to luminance, then they didn't bother with color. Then it was a luminance cell. And basically the only cells they classified as color cells were cells that didn't respond to luminance stimuli, but only to color. And you find about 10% of these cells, but there's many cells, about 40 to 50% in primary and secondary visual cortex that respond to both luminance and color. And we found those, and they were later also found in primary visual cortex by Bob Shapley and his colleagues. So we find no correlation whatsoever between orientation and color. The color cells that we found here are sort of perfectly capable of signaling the orientation of visual stimuli as well. And there's been another study where similar experiments were performed in awake behaving monkeys. We did our recordings in sort of the more classical setup with anesthetized animals. This is from a study of Rüdiger von der Heide and colleagues. Again, the orientation index, how orientation selective is the cell compared to the color index, how color selective. And he recorded from not only from the second visual area, but also from the primary visual cortex, and there's no correlation here whatsoever. So this shows quite strongly that the cells in visual cortex that are selective to color can also code orientation. And you might say, well, how does this go along with other studies? I've read that other people sort of got different results. Well, here's a slide that sort of compares the results from a whole variety of different studies, all done in V2, secondary visual cortex. And the results are also split up. Not only is V2 as a whole being treated, but it's split up into different compartments of V2. There's different subparts of subregions of secondary visual cortex that are sort of more or less concerned with the analysis of color or not. Basically, the prediction here would be that the color selective cells would all be in the thin stripes of V2. So this should be at close to 100%, and there shouldn't be any in the other compartments. And with the orientation, it's sort of the opposite. And what you can see here is that all of these 7 studies are relatively close to the average. There's not that much variation. The studies all more or less agree. Even the ones that are from the most extreme proponents of the different theories. So basically the results that we got, sort of the green ones, are not very different from what Semir-Zeki, for example, had found. It's just that they're typically interpreted in different ways. So while we would say that this is not, there is color selectivity everywhere in V2, Semir-Zeki would say, well, there's much more in these thin stripes here. It's just a matter of how you look at it, whether you say there's sort of a glass of water is half empty or half full. So it's mostly the interpretation that differs. But irrespectively of whether there is sort of anatomical differences what we can clearly say is that at the functional level, there's absolutely no correlation between the two. Now, in these, for the retinal, for the color opponent channels in the retina, I've showed you also some physiology, and then I showed you sort of the cone excitations of real objects and why, based on computational grounds, it's useful to have these color opponent channels. Now I'm going to look, if we have these neurons that are selective to basically oriented colored edges, then those, it would actually, you would assume that those exist in the real world, right? So what we did was, together with Thorsten Hansen, I analyzed sort of a whole large set of calibrated natural images that come from Fred Kingdom's lab. We also used some from the Bristol database. I haven't looked at the ones from Portugal yet. But so we have a whole set of images that are supposed to be more or less typical for our natural environment. And then what we want to look at now is whether the distribution of colored edges in these scenes. And the way we did this was we took the image, like this example image here, and then split it into the luminance and the red and green sub-images, as we know exist in sort of in the retina, those cardinal directions. And then we run an edge detector on these images. In this case, it was just a standard Kenny edge detector from MATLAB, but it doesn't matter. We tried several ones, and you can filter, sort of run it at different resolutions. The results all more or less come out the same. And then we look at the edge, at the combined edge strengths, the edge contrast for the luminance and the colored dimension. So let me show you some examples here. The edge that's labeled number 1 here, it's this transition from red to green. There's basically, this is located up here. It has a large red-green edge contrast and doesn't have any luminance edge contrast. So this is a pure color edge, an isoluminant color edge. Those don't really occur that often, and of course they occur more frequently in this image with this red flowers and green leaves than in any other type of natural image, but they occur occasionally. Edge number 3, which is here, you can see is a pure luminance edge. There's no change in color, only a change in luminance. And as you can see from this diagram, well, those type of edges, number 4 is another case, they exist too. But in many cases, as in this example here, these edges are actually not real, are edges that we might not be that interested in. They might actually lead us astray in our analysis of the visual scene, because it's a pure change, it's a shadow change. So we have a shadow here, and because of the shadow, it's only the intensity that changes and not the color. And of course, if we want to extract the properties of the visual scene, we want to know where the leaves are, where the borders of the leaves are, or where the borders of materials are, and not necessarily, or not only where the shadow edges are. But the important point I'd like to make is that most of the edges are actually have a luminance component and a chromatic component. And when we looked at these histograms for not just this one image, but for all of the images, you can see that basically, I mean, there's some isoluminant edges, there's some luminant edges, but the majority really has both types of contrast. And when we analyzed this also, again, with sort of correlation, and we looked at the conditional probability of having a color edge when there is a luminance edge and the other way around, and we looked at the mutual information. And these color and luminance edges are basically independent of each other. So basically what the visual system should do is to use both types of information and not just ignore one. because the information is there. And typically, whenever our visual system can use some type of information, it typically does it. So that's a good argument, actually, for having these cells in primary and secondary visual cortex that are responsive to color and luminance information. Now, what I've shown you so far was about the color processing in primary and secondary visual cortex, that there are plenty of cells there that respond to color, and most of these respond to luminance and color, and most of these are responsive to edges as well, and they can be used to extract form. And we did lots of psychophysical experiments where we showed that, in fact, our visual system can use that information to extract form. Now, some of the other questions that have been raised a lot about color in the cortex is the question of the color center in the brain. And you might have read something about that there's different visual areas, either here or there, somewhere sort of lateral occipital cortex. If that area is damaged, then people see the world in black and white. They don't see color anymore. Now, that's a bit of a strong claim. There have been cases of patients with achromatopsia where there has been damage, but it's the wrong impression is that those people basically see the world as if on a black and white TV. That's not the case. They have much more damage, and there's one very prominent case that has been studied a lot by Charles Haywood and Alan Cowie. And in that case, basically, the luminance discrimination is just as bad as color discrimination, and still that patient is called sort of colorblind, which is kind of strange. But there's also been new work on functional imaging. And I'm just going to show you one study, because the results of these functional imaging experiments are always, they're actually quite similar. Of course, they're all looking for the color center in the brain. And typically, they find one region in the brain where the color response is slightly higher than in all the other parts of the brain. And that area is then the color center. Now, here in this study, which was done by Doris Sau, Bevel Conway, and Roger Tutel, they actually looked in the monkey brain where sort of we can quickly and easily relate, of course, the anatomical areas to the areas that are known from the electrophysiology. And they also measured the response to color stimuli and the response to black and white stimuli. And basically what you find is that, for example, in primary visual cortex, the color response is much larger than the luminance response. It's almost twice as large. And that has also been shown in many fMRI studies on humans. It always comes out that way. In most other cortical areas, the color response is somewhat bigger than the luminance response. Here, V2, V3, V4, which is sometimes called the color area, and also in inferotemporal cortex, these are also sometimes called the color centers of the brain. Color is always a bit more responsive than luminance, but sort of the biggest difference is really in primary visual cortex. And there's only one area here, the middle temporal area, which is very different. There, luminance dominates. And that's sort of a tiny area that is well established as very important for motion processing. And their electrophysiology has also shown there's not much color there in MT. But if you want to come up with a color center anywhere else, it's sort of very difficult to say which area you would choose. And sort of just from the magnitude of the response, V1 would be the color center. But of course, V1 does everything. And I think so do all the other areas. So there's not much argument in calling one the color center over the others. But of course, there is more to color processing going on in the cortex. So now, before I've shown you, we had these oriented edges. But there's more stuff happening to the color signal. And for that, we and others looked at the responses of individual neurons in primary and secondary visual cortex. Here's again, for comparison, the distribution of preferred colors in the LGN, or the retina. When we had these clusters around the cardinal directions. Now, when the same graph is made in visual cortex, we get a much larger spread of different preferred colors. That is, there's mechanisms sort of tuned to just about any color there. And interestingly, they're not all the same. There are some of these cells that we looked at in area V2 sort of showed a very narrow tuning. So this is sort of the typical response of one of the linear cells as they exist in the LGN or the majority of cells in primary visual cortex. But some of these cells sort of give a very narrowly tuned response here to only particular tones of color. Here's basically I'm showing you what's shown here in a polar diagram is here shown on a Cartesian axis, and you can see here that sort of this widely tuned cell is very linear. The responses follow this sinusoid here, whereas the response of the other cell was very narrowly tuned and basically responded very selectively to this particular reddish tones. And these cells occur basically in secondary visual cortex. There's a few of them in primary visual cortex, but as you go along, they tend to occur more and more frequently. Beville-Conway recently has shown that in parts of infratemporal cortex, these are actually very common. Now, what would you do with cells that are sort of respond just to one particular color? Well, they could be used for analyzing the visual scene. And if you look at the visual scene and objects in the visual scene, I've taken here an image with these colorful hats and some shadows here. And I've plotted a histogram of all the pixels. I've taken each pixel from this image and plotted it in color space. And I've plotted it with the luminance that it occurred on average here. And what you see is that each object basically forms this spike in a certain direction of color space. And now if you want to use these color cells or color luminance cells to segment the scene, then the ideal cell would be one that's sort of tuned very narrowly to a certain direction. And indeed, that's what we seem to have in our brain. Here's another example where sort of the scene is analyzed through sort of a variety of these narrowly tuned cells. And what this shows is that using these narrowly tuned cells, you can segment this image very nicely into the different flowers and into the leaves. We've shown evidence for these narrowly tuned mechanisms that are sort of in a whole variety of different directions of color space, also in psychophysical experiments, but I'm going to skip over those now in the interest of time and get back to our diagram here. So what I've shown you now is this third stage of processing where sort of information from the retina is taken and then combined in many different ways to form these higher order color mechanisms. And how we get from these to these elusive color categories, that's sort of the next step, and it's sort of a quite difficult step. There's been some people who started to look at the distribution of colors in even higher areas of the monkey brain. And recently, Beville-Conway claimed that he had found sort of a distribution similar to the distribution of unique hues that is found in human observers, but it was actually pointed out by John Mullen that that was really caused by an artifact, and we sort of replicated that analysis, and it's really not valid. The other question is, of course, whether, because those mechanisms are very tightly linked to language, whether monkeys even have these sort of unique hues or these categories the same way as we do. Okay, so looking at this picture that I presented in the beginning, I've showed you now approximately what's going on here, but of course the whole thing is much more complicated, and there's many other interactions of color with other parts of the visual not only of the visual cortex. For example, memory can affect the way we perceive color, so-called memory colors. If you look at the scene of fruits again, and you always have the impression that the banana is yellow and the orange is orange, but sort of when I take the context away, you can see here very nicely that the color actually changes quite a bit from here, from reddish here to orange to greenish here, and still we always see this as nice orange. And of course, all the typical mechanisms of color constancy play a big role here, but recently we were also able to show that our knowledge about the color of objects can have an effect on our perception of the world here. Now, to summarize what I've shown you, color is processed very efficiently in the retina. And color and form are processed together in the visual cortex, tightly linked, and there are specific color-tuned mechanisms in visual cortex, the so-called higher order color mechanisms, and these mechanisms contribute to image segmentation. And lastly, I'd like to thank the various current and previous members of my lab and the Deutsche Forschungsgemeinschaft for funding all this research for many years. Particularly, I'd like to thank Maria Olkkonen, who's from Finland, and a very promising young color vision scientist. Thank you very much.
Thank you, Professor David. Now, questions or comments or argumentation or...
Carl, thank you for a splendid review of an awfully complicated area. You've made a case for essentially a disconnect between function and structure, loosely speaking. Would you have expected more evidence of structure reflecting function in the early levels of the visual cortex than you've actually found?
The disconnect you're referring to is...
Well, for example, the lack of correlation between orientation and color, and so on.
So there, yes, so there are distinct functional streams in early visual cortex. That's what you're referring to. And basically, I think this is sort of, as you know, in the retina, there's this three different channels, sort of the magnocellular, parvocellular, and more recently the coniocellular layers have been found, and it's known that these work independently and convey information to visual cortex. And sort of the very earliest implementation of this coloring book theory, as I call it, or you might call it segregation of color and form theory, sort of just claimed that the segregation between magno and parvo is just going through all the way in visual cortex, and then there's a sort of a dorsal stream in visual cortex where movement is analyzed that gets all its input from the magnocellular layers of the retina, and a ventral stream that gets all its inputs sort of from the parvocellular layers. Now, it's even on anatomical grounds, it's now quite clear that this is not the case. There is one tiny little area in the brain where it really seems to work like that, in that this motion area MT seems to get 90% of its input from magnocellular parts of the retina and the geniculate. But that's really a very, very small area. But all of the other areas seem, the magno and parvocellular signals are already mixed in primary visual cortex. They end in different layers in V1, but then they're immediately sort of intermingled. And but V4, for example, receives about half magnocellular and half parvocellular input. So in the first anatomical experiments that were done, it was really this sort of segregation that was put forward very clearly. But in later experiments, mostly by Sinkic and Horton, they found that there's lots of interactions between these different streams as they pass through visual cortex. So I would say there might be a little bit of segregation left, but it's at it's true. I mean, anatomically there is a bit of segregation left, but functionally it doesn't seem to play any role anymore.
This doesn't worry.
It doesn't worry me.
Okay, next question.
Just would like to know your opinion about the idea that evidences tell us that color helps with edges, but maybe edges helps with color as well.
Well, if you have a mechanism in the brain or a neuron that responds whenever you have an edge with a certain color and luminance contrast, then you cannot really say, I mean, then you can always say, well, the edges help the color and the color help the edges, but the two are just put together. I mean, you cannot separate it. It's one and the same. That's the way I would see it. In terms of the edges help color, I mean, you might refer psychophysically to sort of filling in phenomena where sort of colors can propagate from the edges. That would certainly also be a... But I think that this filling in is a phenomenon that works at a different level. Yeah, I mean, that's true. Of course, the cone contrast across edges can be extremely useful for color constancy. I mean, that David has shown very clearly.
I have some kind of unusual question about ethics and your research. So where does information come from when you're talking about monkey brains? I know already when we do psychovisual experiments with humans, it takes a lot of time. It's some kind of inhuman sometimes. So what about information from animals?
Well, I mean, the monkey has been used a lot in physiology experiments because the visual system of these macaque monkeys is thought to be quite similar to that of humans. It's now clear, sort of comparing, especially functional imaging experiments in the monkey brain and in the human brain, that of course, as the higher the level you go, the more distinct the two become. Not surprisingly. But I think at this early level of primary visual cortex, our brain is probably still quite similar to that of the monkeys. But did I understand your question correctly?
The moral aspect.
Oh, the moral aspect. Well, I think that will be a discussion that should probably take place somewhere else. But I think it can be stated that everything we've learned about cures of the visual system, we've learned from these animal experiments. And we're sort of far from the point where functional imaging would give us any sort of new insights that would help us in curing diseases of the visual system.